Good morning, everyone. It is Tuesday and January the 12th. It's 9 a.m. here in Cleveland, Eastern Standard Time. My name is Ashok Agarwal. I'm your host for the next 2-3 hours. And I want to, first of all, before we begin the program, want to wish you a very happy New Year 2021. I wish you good health and a lot of happiness, and I hope that we will be able to see many of you during this year after the pandemic is under control. So thank you again. And today is a very exciting program. We are here to discuss and present virtually our online ART training. Today is the third program in the series. We began in November. We presented first program in November, then second one in December, on December 8th. And today is our third program on January the 12th. Today's program is on Ancillary Semen Tests. So let me begin without taking any further time and begin my introduction. Very briefly, this is my biography, a very brief biography, just to provide few highlights. I am in charge of the Andrology Center at Cleveland Clinic for the past 28 years. I'm the director of the American Center for Reproductive Medicine, which is the host of this online virtual training session. I'm a professor of surgery and urology at the Lerner College of Medicine at Case Western. I'm a staff in urology and in some other department. Our group is very well known in the area of male infertility, andrology, and human-assisted reproduction, and we are ranked as #1 in terms of our publications based on the Scopus, which is one of the premier service in terms of the literature in the medical field. Our group has published a very large number of books in the field of male infertility, ART, fertility preservation, DNA damage, and antioxidant. Our laboratory has trained over 3,000 scientists, clinicians, graduates from more than 100 countries. The organizers and speakers of this program have no conflict of interest. We have taken no sponsorship or asked any company for that. This program is made entirely possible by the support from our foundation, from our own center, and the webcasting support has been provided by our colleagues, David Reichling and David Wolfley. Course faculty have generously donated their time for this online training, and our program coordinators, who you will see in a minute, have graciously volunteered their valuable time to manage this program. Lastly, the names, images, and description of products or instruments that you may see during the lectures or which will be discussed during the presentations are not being endorsed. They are being used routinely in our program. So this is today's program. The learning objectives today is we have a very, very good program today, and you will see this is part of the semen analysis, and it is like a hidden gem. Most people will not know many of these tests that are basically done as part of semen analysis as indicated. So we are going to discuss about the leukocytospermia or detection of white blood cell or infection, sperm vitality testing, anti-sperm antibody screening, special test for azoospermia, post-vasectomy screen, and screening for retrograde ejaculate. So I can tell you that today's program is by far going to be the very best. We have invested enormous amount of time in preparing for these lectures. They have been well researched, well prepared, and practiced dozens and dozens of time what you are going to see today. So for each lecture, we have spent almost 30, 40 hours minimum in practice sessions. And each lecture has gone through at least 10 or 12 practice sessions with multiple people. And each content has been well researched through the literature with proper references, and to make sure everything is extremely accurate and simple for you to understand. So this is our program today in a diagram. As you can see, the last lecture is going to be by our clinician, who's going to really put it all together to explain what these tests really mean to you. Okay, our first speaker is Professor Ralf Henkel. He is a highly decorated researcher, academician, and a teacher, all rolled into one. And we have been very, very good friends, but he is listening to us, and he has recorded his presentation just to avoid any kind of technical problem. But let me come back to his biography, which is very, very impressive. As you can see, currently he is a visiting reader in the Department of Metabolism, Digestion and Reproduction, a mouthful, in the Imperial College of London in the United Kingdom. He is also the chief scientific advisor at a very important company called Logix Pharma in Reading, United Kingdom. He essentially moved from South Africa, where he is currently an extraordinary professor of reproductive biology at the Department of Medical Biosciences, and in a host of other places. He is also affiliated with our program for many, many years. We are working together. Most importantly, Ralf is the editor-in-chief of Endologia, one of the original and most prestigious journal in the field of andrology in the world. He is very well recognized in the world, and you can see all his information in the slide that we will later on share with you. So we are going to play his presentation as a video, although Professor Henkel is online attending this program, but he has some technical issues, so he was afraid that it can create a problem. So he preferred that we play the program through the video that he had kindly sent and recorded beforehand. So we are going to play it now. So his lecture is on leukocytospermia.
And good morning, good afternoon, good evening, everybody, and welcome to my presentation on leukocytospermia today. The learning objectives today are to understand the need to examine the presence of round cells in semen, testing the presence of white blood cells in an ejaculate, defining a leukocytospermia condition, and how to test for leukocytospermia. And of course, when we test clinically, when we need quality control, and we also need to understand the relationship between leukocytes and the infection. What is a leukocytospermia? Leukocytospermia is defined according to WHO in the latest manual of 2010 as a white blood cell number of more or equal 1 million white blood cells per milliliter of semen. This is a condition that is present in 10 to 20% of infertile men. And it is an indication of male accessory gland infections, or abbreviated MAGI. And this is or can be seen in up to 15% of male infertility cases. Where does these infections take place? That is mainly in the testis as an orchitis, in the epididymis as an epididymitis, prostate as a prostatitis, or in the seminal vesicles as vesiculitis. And that goes along with a swelling of the relevant organs here, and real infection of these organs. It's getting along with fever, temperature, heat of the relevant organs. It's painful, and it's due to a number of pathogens like chlamydia, ureaplasma, mycoplasms, enterococcus, staphylococcus, and pseudomonas. The detection of these conditions is difficult because in many cases, these patients are asymptomatic. We also call that as a silent genital tract infection. And we need to understand that these MAGI, or these male accessory gland infections, they contribute to increased seminal leukocytes. And that is in the end leading to increased production of reactive oxygen species, and as a result of that, to failed fertilization and pregnancy. And then we have problems with treatment of the antibiotics, because many of these pathogens in recent years became resistant to antibiotics. And that must then be tested in a semen culture where the different possibilities can be checked for the sensitivity to different antibiotics. Also, there are different types of pathogens available, aerobic and anaerobic, and both aerobic and anaerobic pathogens are present in semen, with anaerobic pathogens even in almost every ejaculate available. In addition, there are different other challenges that need to be observed. There is no agreement and no guidelines from the WHO, from the AUA, ACRM, or from the European Association of Urology to give guidance when a semen culture should be done. In addition, there is the relevance and significance of the positive semen culture is debated, but on the other hand, it's critical to distinguish between semen infections and to know what is going on there for an effective management and the way to treat these patients so that we know what can be done. In this recent study of last year by Burry, these authors investigated more than 1,600 patients, and among these patients, they found asymptomatic, so with patients with a silent infection, and a positive semen culture that was 21%. It's a substantial number, and most of these pathogens that were found was ureaplasma, ureolyticum, and there was enterobacteria, chlamydia, mycoplasms, and others, even here including a virus. And in more than 10%, there was even more than one agent, one pathogen detected in the semen. As a result of all this study, these authors could clearly see significant differences for sperm count, progressive motility, and more between positive ejaculates with the infection and those that are not infected. In another study, Ventimiglia, also from last year, these authors investigated more than 500 patients in 90% of these patients, the semen culture was negative. However, in 10%, the semen culture was positive. And if they analyzed these 10% further, only 20% of these patients were positive for leukocytospermia. That means that 80% of these patients would not have been detected if they would just have relied on a leukocytospermia test and would not have done a semen culture. So that means that leukocytospermia is not necessarily a good predictor of positive semen culture, and it's no single parameter is sufficient in medical decision making, because we need to treat the patients properly and effectively in the end. What kind of leukocytes will we find in semen? It's the majority of the leukocytes in semen are granulocytes. That makes between 15 and 60%, which is neutrophils or polymorphonuclear granulocytes. Then there's about 25 or 20 to 30% of macrophages, and the rest is then T and B lymphocytes. And the detection of these leukocytes in semen, there's different methods available. We can do it with immunocyte staining chemistry, that is regarded as a gold standard with monoclonal antibodies against specific white blood cell subpopulations. And this can also be done in flow cytometry, as shown here on the right-hand side. Can clearly distinguish here this group of leukocytes, and here at the bottom there are the spermatozoa. On the other hand, there's specific tests available, like seminal granulocyte elastase test, and this is done in an immunoassay, ELISA. But all these tests have limitations. They are, number one, expensive, they are time-consuming, and they are not standardized. So what can be done? There is routine tests that give an indication what is going on in the ejaculate. In a wet prep, one can see round cells, but these cells cannot be really distinguished from immature germ cells, and it's highly inaccurate. So we need to do other techniques, including staining techniques with Papa Nicolau or modified Bryan-Leishman staining. But again, these techniques are time-consuming, require really very specifically trained staff in the laboratory, and they are not precise. And it's a problem to distinguish between different germ cells and different round cells and leukocytes. So what is left, and also been recommended by the WHO, that is the Ortho-Toluidine blue test and the Endtz test. What we see if we look in the ejaculate in a normal light microscope with our round cells, that is for the initial semen analysis in the wet prep. And what we can also see that we cannot distinguish between leukocytes and immature germ cells. There is even other cells available, epithelial cells of trichomonas, and these cells here are primary spermatocytes. Here is the spermatid, here even the trichomonas. You cannot really distinguish between these specific cells and is it active leukocytes. So we need to get down to the bottom and clearly identify leukocytes. And but this examination for round cells, that is a first indication. And if there are more than 5 round cells per high power field, then we need to check for white blood cells or leukocytes. If we see more than this white blood cells, and it should be done according to this formula, we count in a light microscope all these 10 by 10 grids, and it's more or equal than 1.0 million leukocytes per milliliter, then we have to do the leukocyte test. And there are two different leukocyte tests available. That's an Endtz test or the peroxidase positive cells test with the Ortho-Toluidine. Like I said, that test should be done if there is more or equal than 1 million round cells per milliliter ejaculate, when either Endtz test or peroxidase test. The Ortho-Toluidine peroxidase test is recommended by the WHO. We need to prepare all these chemicals, reagents, working solutions. That is when must be done on a daily basis. After staining procedure, what is shown here, when we see in the microscope peroxidase positive cells, that is those leukocytes that still have the peroxisomes in the cell, they are staining brown like these cells here, and those that are negative are not leukocytes, that is these cells here, they are markedly lighter, more whitish. On the other hand, there is the Endtz test available, which is named according to the person who first described this test in 1974. We also need to prepare all these stock solutions which are stable for about 6 months, and then the working solution should be prepared on a weekly basis. The test is then performed with 20 microliters of the Endtz reagent, with 10 microliters of thyroid buffer or PBS buffer and 10 microliters of liquefied semen, which is mixed in an Eppendorf cup, and that must then be a dark brownish one to prevent it from exposure to light. After the incubation, we can analyze it in a macular chamber under a bright field microscope, count the peroxidase positive cells, and do the calculation. And this slide shows you the performance of the Endtz test and all the equipment that you will need. And now I'm going to show you a small little video about the performance of the test. Here you take the 20 microliters of Endtz solution in a dark Eppendorf cup. You add the PBS of the solution or thyroids, mix carefully, and then add the working solution. Mix again, and then incubate for 5 minutes. Afterwards, you prepare your microscope in the bright field. Maybe take away the filter. It is used with a 10 times objective lens. The macular chamber will be filled with the exposed and the incubated solution with the sperm. Okay. Now the cover slip comes on top, and we can then analyze this in the microscope. Of course, for any normal diagnostic test, we need to do the quality control, and that should be done on a weekly basis for the Endtz test. The Endtz positive control should be run, and the result should be more than or equal 1 million white blood cells per milliliter. If the results are less, then prepare new reagents and retest the control. If there's no leukocytospermic sample available, one can also use EDTA anticoagulated blood specimen. And here on the right-hand side, I'm just showing you some examples of a quality control sheet. Here you can see the sample that was done last year in September, 7.6 million leukocytes per mil in the old solution, and the same here for the new solution. So that is perfectly correct, and there is no difference between these. So that quality control test is positive, and that's in order. What kind of leukocytes can we detect with the Endtz test? We detect neutrophils, eosinophils, and basophils, but we cannot distinguish between these three different types of granulocytes. Again, if the result shows less than 1 million leukocytes per milliliter, that is then not leukocytospermic. If it's positive, it's more or equal 1 million leukocytes per milliliter of semen. Where are these reactive oxygen species coming from in semen? That is, of course, here from white blood cells. That is their normal function, what they are doing to actually attack pathogens and destroy pathogens. On the other hand, they are also coming from abnormal spermatozoa. And there we must also be aware of the fact that the activated leukocytes, like here, they are producing roughly 1,000 times more reactive oxygen species than normal spermatozoa. And how is that done? In abnormal sperm and in leukocytes, they follow basically the same biochemical pathways, where when apoptosis is activated and oxidative stress via reactive oxygen species and reactive nitrogen species are released, which in the end damage the DNA, proteins, and the lipid membranes of good quality spermatozoa, leading to a reduced fertility of a specific patient. If we have leukocytes in a semen, that is an indication there is also more reactive oxygen species produced and released, and this is clearly, you can clearly see here in this study, Agarwal in 2014, leukocytospermic samples, the production of reactive oxygen species is significantly higher than in semen samples of non-leukocytes or even in non-leukocytospermic patients. This elevated level of leukocytes has also a significant effect on sperm motility. So motility goes down in semen samples that contain higher numbers of leukocytes. As well, here, this study, the amount of DNA damage is increasing significantly with elevated numbers of leukocytes. And here in leukocytospermic patients, it's also significantly higher in this other study by Saleh et al. And with this, I want to provide the take-home message. The peroxidase staining by Endtz test, or the toluidine blue stain, while it's not a gold standard, it's simple, it's practical, and it provides a reliable result. Leukocytospermia is called when there's more or equal than 1 million of white blood cells per milliliter of semen. It may indicate an underlying genital tract infection in terms of MAGI, and leukocytospermia is not a good predictor of a positive semen culture. That's why both tests, the leukocytospermia, the detection of leukocytes with the Endtz test also, and the semen culture should be done in parallel. And leukocytospermia is a significant factor associated with high reactive oxygen species, resulting in poor sperm quality and DNA damage. And with this, I thank you very much for your attention. Here are some pictures from our group. And this is where I'm staying at the moment in London, Hammersmith Hospital.
You can connect with our program by connecting on our LinkedIn, and these are the benefits. Almost every single day, we provide update on our research activities by posting hundreds of research articles freely, completely downloadable, that anybody can read. We also post the latest research which are presented at prestigious meetings such as ACRM, Accessory, and AUA. You can learn about our virtual research opportunities, which we have just started. You can learn about our online training and opportunities to collaborate with our team. Just go to the LinkedIn and send a request to Ashok Agrawal, and it will, from Cleveland, and it will come to us, and we will allow you to connect to our program. This is actually the best way to learn all the advances that are happening.